I'm Mark Banish. I'm President and Chief Scientist of Neotericon, LLC, a nanotechnology and nanomaterials consultancy located here in Bedford, New Hampshire, USA. If you'd like to contact me after this is over with any questions, please do so. The emails are at the bottom. Today, I'd like to talk about something which is extremely important with carbon nanomaterials, namely the presence of what are called polyaromatic hydrocarbons, and how you can measure and analyze for these using a remarkably simple technique. So what are PAHs? Well, PAHs are made from rings of 6 carbon atoms arranged as a hexagon. Now, there's something very special about this building block, and that is that the bonds between the carbon atoms, well, there are some that are fixed, and then there are some where the electrons are free to move around the ring in something called delocalization. Now, this is unique to these types of structures, and the good thing about that is that the delocalization makes the structure very stable and resistant to chemical reaction once it's formed. This enables the molecules to survive in all sorts of tough environments, even deep space. If you take a look on this slide, you'll notice that there's a collection of PAHs, all sorts of hexagonal rings, all that have been found out in deep space by NASA using spectrographic analysis of lines, emission lines, from places like the Iris Nebula, which is over 1,100 light years from Earth. So they're easily formed in nature, and they're found in some of the most unusual places. One PAH of particular interest, or something that's related to them at any rate, you can see in the lower side of the diagram. It's a buckyball. That's because the chemical conditions that give rise to PAHs are often the same that give rise to nanotubes, or at least allow nanotubes to grow. And the same thing for fullerenes and carbon nanofibers. So this is why we want to study PAHs. It's because they are chemically linked to nanostructures, carbon nanostructures. Now, they can form as side reactions, or they can be called contaminants. And in some instances, they can make an appreciable amount of the end product. They can be 5 to 10% of the mass. And since they have environmental health and safety concerns, they're carcinogenic, for example, and because when they land on the surface of a CNT, they can block sites on the CNT from interactions with something, say, like a composite matrix, well, then they can affect end performance as well. So understanding something about PAHs, specifically, at least in the beginning, how to measure them, how to detect them, quantify them, is very important in understanding how nanomaterials work, or at least nanomaterial products work, and what obstacles they may have to getting them to work successfully. So how do you get rid of these things? Or better yet, how do you extract them and isolate them for study? And the answer is with a simple device called a Soxhlet extractor. Now, this is really just a condenser, but it's a very special kind of condenser. It uses a solvent reservoir, which is heated, and the solvent is heated to boiling, and the solvent vapor then goes up the sidearm of the condenser, and it then hits a cooling water coil and condenses back down as pure solvent, and that flushes over your sample. The nice thing here is that you're always flushing with fresh solvent because the solvent vapor that comes up from the reservoir is always pure. So this ensures that whatever's in there is going to get a pretty good washing, and you can run this unattended. It requires some specialized glassware, but it's not really that expensive. And while the technique itself, in theory, sounds very good and very simple, there are some restrictions with respect to polyaromatic hydrocarbons. First, you have to have a solvent which dissolves them. And this kind of limits you from things which might be more benign chemically, at least to humans. Also, you want that to be removable from the analyte. That is, that if you want to measure the mass before and after to determine exactly how much of the material came off, you don't want something sticking there and just simply taking its place. And this excludes certain solvents, such as toluene and xylene, which bond very strongly, or at least strongly adsorb, onto the surface of things like nanotubes, fullerenes, and carbon nanofibers. You want it to be compatible with any subsequent analytical technique. And this leaves you with two, well, acceptable choices, and that would be chloroform and methylene chloride. Now, of course, these aren't necessarily first choices you might have in terms of them being relatively benign, but under the proper circumstances and with the proper supervision, they can be used safely. You can use something called Hansen's solubility parameters if you want to find others, but methylene chloride and chloroform are two good choices to get started. So here's the methodology. You need a reasonably sized sample, about 100 to 500 milligrams. Put that in the Soxhlet extractor, add about 150 milliliters of your solvent, and extract over and over and over. And eventually you take an aliquot of the solvent that's inside the Soxhlet extractor. And since PEHs fluoresce in the ultraviolet, you can use UV spectroscopy to monitor how pure that solvent is. And when you're seeing nothing but pure solvent in there, because everything's been dissolved, well, that's pretty much time to stop the extraction. You remove your sample. You can concentrate it if you'd like. You can concentrate that collected solution, which is drained into the reservoir. You can leave it alone, depends on however you want, but you have to weigh the analyte before and after to know how much material came off. And you then are going to use a standard piece of equipment, a gas chromatograph mass spectrometer, GCMS, and by putting in some control solutions of known concentrations, it's easy to determine the response of the instrument and be able to determine the quantities actually present in the solution being analyzed. And here's an example of an actual material that's sold on the market. It's called 2-ball 75% single wall nanotube by mass. And you'll see here is the results of the analysis of the Soxhlet extracted, in this case, it was dichloromethane. You can see that the materials that are pulled off are naphthalene, acetonaphthalene, two very simple polyaromatic hydrocarbons. Pyrene is also present. Now, the interesting thing here is that naphthalene, which is probably the simplest polyaromatic hydrocarbon, forms about 90% of what we pull off, while the pyrene is a little under 10%. But you'll notice that the amount extracted is around 7%. So basically 7% of the mass of this product is actually not nanotubes at all. It's polyaromatics, and it's mostly naphthalene. And that is all absorbed onto the surface of this material, and it is all so thick at this level, you've probably got more than a monolayer. It may even be encapsulating all of the nanotubes. So essentially what you have is a nanotube covered by a polyaromatic hydrocarbon. So this is just a quick overview of how this works. I am working with the ISO Technical Advisory Group to turn this into a standard, and eventually we will have a more complete, detailed procedure. But I think this gives an excellent sense, an excellent flavor to how you can go about analyzing these things. And you can see that they are there. Now, one last thing that I'll put here is how do I know those signals that I see in the GCMS are what I think they are, that they're naphthalene, pyrene, or whatever? And the answer is I am very thankful to NIST in the fact that they have now made their database of mass spectra of polyaromatics available. And for most GCMSs, they are accessible as part of the software package that drives the instrument. So it's really relatively straightforward to make sure that you have what you have by using a trusted reference from a trusted source. So this is just a quick overview of how you measure PAHs in carbon nanomaterials. And if you have any questions, my email's there at the beginning. And thank you very much.